Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Good morning everyone. Welcome back to my channel Pari's tutorial. Let's start the class. In the last class we have already discussed about the protein separation by STS and DNA RNA separation by agarose gel electrophoresis. Now my topic today is about the blotting techniques. Okay, what are the blotting techniques? So why are the blotting techniques used? The blotting techniques are meant for the separation of the particular DNA molecule or particular peptide sequence in protein. On STS electrophoresis, you got the sample of proteins, but you want that particular peptide sequence of your interest. So for that, you have to go for blotting technique, where they will use a probe that was being labeled with enzyme or any radioactive active labeled thing. Okay, so let's start. What are blotting techniques? So blotting is a process of transferring the macromolecules like proteins, nucleic acids, etc. from gel to the solid surface of an immobilized membrane to detect the molecules. Okay, so as I told you, we'll be getting a gel where the bands have already been produced. That should be transferred to a nitrocellulose membrane for better analysis. Okay, so this is done by either capillary method or by electrofusion, like electric current passage. Okay, so I'll tell you what are the different types of blotting techniques, then you'll get an idea of how they are used and when they are used. Okay, so why nitrocellulose membrane is used? Nitrocellulose membranes are considered to be a popular matrix for the protein binding. It has got the detection. Okay, that is why nitrocellulose membrane is used. It has the ability to immobilize those particular proteins, that protein of interest, whatever we are targeting, that can be easily mobilized on nitrocellulose membrane. So for that, what are the types of blotting? So there are mainly of three types: western blotting, northern blotting and southern blotting. Western blotting is meant for detection of proteins, northern blotting is meant for detection of mRNA, and southern blotting is meant for detection of DNA. Okay, let's start with southern technique. So it was first developed by Southern in 1975. Okay, so I told you that southern blot is meant for the detection of that particular fragment of DNA in that sample. Okay, so DNA that is detected can be a single gene or it could be the large piece of DNA from a viral genome. Okay, it could be a part of it or it could be a complete one. So that should be of our interest. That has to be separated by blotting technique. Okay, see what is the principle involved in it. The samples have to be run on agarose gel electrophoresis. Then you will get a gel, and gel has to be transferred to a nitrocellulose membrane, and then you can go for further X-ray analysis. Okay, see I will tell you what happened. So when you just immobilize it, like the DNA sample is being immobilized, now you will add a probe. So probe is something that has the same sequence that is complementary or it has binding capacity with the DNA of your interest. If DNA has got a homologous to this part of DNA, so that it could be easily binded to it. So that probe should be attached or should be labeled with an enzyme or a radio labeled substance attached to the target DNA molecule. Now the remaining unbound probes will be easily removed later on. Okay, in the process you will be removing it later. So once the unbound, the bound one can be further gone for chemiluminescence, where you can see the darker bands of your target. Steps involved: digest the DNA with appropriate restriction enzyme. Complete complex mixture of fragments is subjected to gel electrophoresis, as I told you, and the whole these are the steps that are involved in southern blotting. Okay, so this is how the capillary action of the gel is transferred to nitrocellulose membrane. So the gel is placed with membrane and sheets filter paper, tissues and weight, so that with capillary mode of action, the gel gets transferred to the membrane. Okay, so what are the applications? So southern blots are mainly used for gene discovery, mapping, DNA fingerprinting also. Okay, so these are the important points to be covered in southern blotting. So I told you the next one is northern blotting technique. What is northern? So it is also called as an RNA blot. So because RNA is a important genetic material for many viruses also, right? So RNA has got more susceptibility for the degradation than the DNA. To be loaded on the gel, and RNA samples are separated according to agarose gel electrophorese. Okay, gel is again blotted on the linol membrane or in nitrocellulose filter paper by sandwich arrangement. As I already told you, it's the same process here. Okay, so that continues. This RNA is then immobilized on the membrane either by baking at high temperature. So I told you that nitrocellulose membrane has got a good affinity towards proteins and nucleic acids. This RNA would be easily immobilized either at high temperatures or by UV cross linking, which results in the covalent linkage. I told you that RNA gets linked covalently with the nitrocellulose membrane so that it won't get washed away for the subsequent processing. Okay, so this is followed with a DNA probe or RNA probe. So when you want to know about the RNA, then you can either add the DNA which has got the homologous sequence of your target RNA. So when you add this, you will be getting a double stranded DNA, whereas one strand is DNA and other stranded is RNA. It's a hybrid, right? So that particular hybrid can be easily visualized as a dark band in X-ray film. Okay, so that is how you will hybridize it. It contains complementary RNA sequence. The probe will bind to the membrane to form a double-stranded DNA and RNA hybrid molecule. Okay, so you already know that remaining things can be easily washed away with the unbound probe. Okay, now the labeled probe is detected via autoradiography or by chemiluminescence. So in both the cases, you will be getting a dark band in X-ray film. So this is how the technique is. So I told you RNA has to be separated, should be run on agarose gel electrophoresis. Once you get the gel, you have to separate it and place it on the nitrocellulose membrane, and again it is also a capillary mode of action like southern blotting also. Okay, so again after that you will be getting concentrated DNA, RNA bands here, and you will radio label them with the suitable probe, and finally you get a band with the target of interest where the concentration is higher. Okay, this says that this particular sample has got a higher target interest because more probes got attached to the sample of this gel. In this sample, your target RNA is more in this sample. That is why the more radio label probes got attached there, and it is clearly shown in X-ray. So this is how you analyze the blotting techniques. So northern and southern follow the same process, but the western blotting is something different. So applications, I told you, these are meant for studying of RNA degradation, study of RNA splicing, and RNA half-life can be detected. Okay, so these are the main applications of northern blotting. So western blotting, this is considered to be also called as an immunoblot because you have already the undergraduate students would also would be having some idea what is immunology is, right? So it's all about immune systems and all. So where you have come across the antigen antibody interactions, I will cover this immunology in the last or either in some other class because immunology is very important topic. In that technique you will keep hearing this western blotting technique a lot. This western blotting is also called as immunoblotting technique, where you will specifically detect that particular protein sequence or amino acid sequence by blotting technique. Here the blots are to be transferred through electrofusion, like electric fusion, not by capillary action. Okay, so this is also called as immunoblotting, and here you don't label it with any radioactive, but you will be reading with a secondary antibody. So you know that antigen antibody interactions are like a lock and key model, like it's like enzyme substrate reactions, like they are all bound to each other like lock and key. So that's how the main principle involved in western blotting is when you get a sample, when the nitrocellulose membrane has got a protein of interest, you will get bands based on it. Okay, so let's go for process what really happens. So in western blotting, you will be meant for western blotting is meant for the separation of proteins. So for the proteins, you all know STS page is the condition to be western blotting. So STS is a sodium duodecyl sulfate polyacrylamide gel electrophoresis, where the proteins are to be first separated based on the molecular weight, right? So once they are done, they have to go for the western blotting technique. In the western blotting technique, you will get a gel based on the bands and all, you will detect the molecular weight, and now this electroblotting is to be done. Transfer the separate proteins from the gel into the nitrocellulose membrane. I told you it's electroblotting in western blotting, it's electroblotting. Okay, the blot is incubated with generic proteins such as milk plug or BSA, bovine serum albumin, okay, which binds to any remaining sticky places on the nitrocellulose membrane. You all know that important principle of western blotting is the proteins of that, or that particular target protein, it should be added with a suitable antibody, right? That is what you call as primary antibody. You will get a complex, like if it is antigen here and you have added the antibody. Now if you think this is the nitrocellulose membrane and you got a small target antigen there, right? So for that you have added the antibody, which is called a primary antibody suitable for that. But to this primary antibody you will be adding a secondary antibody which was being labeled with enzyme, right? So what you do is this secondary antibody with enzyme linked to it will be added again, so that this secondary antibody would directly bind to the primary antibody and forms antigen antibody complex. The whole thing is done. You will be adding a suitable substrate, because you know enzyme substrate is like a lock and key model. So for each and every enzyme a suitable substrate has to be added. So when you add the suitable substrate for the enzyme that you have linked, if they give a separate band on the X-ray that says that that protein molecule has got the target protein that you are thinking of or you are calculating of. Okay, see, an antibody that is specific for the protein of interest is added to the nitrocellulose sheet and reacts with the antigen. Only the band containing the protein of interest binds to the antibody, forming a layer of antibody molecules. I told you. After washing for the removal of non-specifically bound antibody 1, secondary antibody is added which specifically recognizes the FC region. Yes, I told you each and every antibody has got FAG, antigen binding site and domain site. Okay, this I will tell you what are the types of immunoglobulins. I will explain with the structure also in immunology class. Okay, as of now, please remember that the FC domain will be recognizing that particular antibody, primary antibody, and finds it. So this antibody, secondary antibody is now radioactive labeled and is covalently linked to the enzyme. Now you will get a complex, as I told you. So this is the easiest diagram that I can explain you. So this is gel, and this gel now is made to electro transferred. Okay, electro transfer has been done and it is moved to cellular nitrocellulose membrane, and there you got different bands. And see, in this sample the primary antibody is added, right? This is the primary antibody suitable for your protein of interest. And now you have washed the remaining unspecific non-specific bands, non-specific things are all removed off, and then you are added the secondary antibody that was being labeled with a enzyme substrate, like it's a enzyme related, enzyme linked. Okay, and this enzyme linked would, this secondary antibody with the enzyme linked would be binding to a primary antibody, forming an antigen antibody complex. So after forming the complex, now you will be adding for the substrate. When you add the substrate, you will get the film in the X-ray film where you can see the bands here. See, this is a higher band. This says the concentration of your the protein concentration is more here, because this is how you analyze the Western blotting technique for the separation and identification of your proteins. Okay, so these are the blotting techniques and application. This is a common questions asked. It is a confirmatory test for HIV. It's a definitive test for bovine spongiform encephalopathy, and some Lyme disease can also be detected by Western blotting. And this is a common question. It is a confirmatory test for human immunodeficiency virus. Okay. So these are the differences. If you like to get into a simple way, this is how you make it as brief as possible. Okay, so that I told you, you can please go through the slides once again if you have doubts. Okay, please go through the channel and get to know the difference between all the blots. Okay, it is blotting techniques. Okay, in the next class I will teach about the separation of proteins and analyzing the proteins by mass spectrometer, because that is the last class for the separation techniques. As I have already covered most of the separation techniques, I would end up with the mass spectrometer. Okay, chalo, thank you so much. Please like, share and subscribe my channel. See you in the next class. Thank you. I have attached the links for the topics in the description box. Please check it out. I have attached the links for the topics in the description box. Please check it out.